I'm going to be talking to you about the uses of genetic data in psychology. Now, it turns out that psychology already has a subfield that has traditionally made a lot of use of genetic data. This subfield is called behavioral genetics, and its practitioners study the inheritance of mental abilities, personality traits such as how outgoing you are and so on, vocational and recreational interests, religiosity, political beliefs as we heard about, mental illnesses such as schizophrenia and autism, and also perhaps surprisingly fertility, which was recognized by Ronald Fisher long ago to be as much of a behavioral trait in humans as it is a physiological one. Now, talk about abilities and personality and so forth is fairly coarse-grained. At a finer grain, behavioral genetics aspires to address how the genome encodes a brain capable of more elemental cognitive feats such as seeing, thinking, talking, moving, and so on. Now I'm going to give some reasons why I believe behavioral genetics is a scientifically worthwhile endeavor. I start with the observation that a typical human brain consists of roughly 100 billion neurons. Neurons are the information processing cells in your brain as opposed to other cells such as glial cells and so on. And also at least 100 trillion connections between these cells, which are called synapses. I say at least because these things, even with modern technology, are still very difficult to count. It may well be that the number of synapses in a typical human brain is an order of magnitude more than this. But nevertheless, these are still very large numbers. In fact, this number of neurons is roughly equal to the number of stars in the Milky Way galaxy. And these neurons interact with each other in a manner far more complex than the mutual gravitational influences that give the galaxy its overall structure. So we can forget about the three-body problem. This is something that would have given Newton headaches. Now, one thing that's remarkable about this is that all of this complexity flows from a genome that consists of only roughly 100 megabytes. So the precise number depends on certain things that we're not quite sure of yet, such as what percentage of the genome is actually functional. But this is definitely within the ballpark. And to many people, this seems like a surprisingly small number. I mean, I have several GWAS meta-analysis files on my hard disk that are each bigger than 100 megabytes. And so this disparity in magnitudes leads to an important question. How is it that such sparse data is capable of encoding a device of such enormous complexity? So another motivation for studying behavioral genetics is the so-called nature-nurture issue, a euphonious name for this opposition that was coined apparently by multiple writers at almost the same time in Shakespeare's England. So these famous lines from Shakespeare's final play that he wrote all by himself, The Tempest, these are spoken by the magician Prospero when he's asked to explain the character of the villain Caliban. Later in the play, when Shakespeare gives Caliban a chance to speak for himself, he, understandably perhaps, has a different opinion. But never, regardless of who's right, I think the important point that emerges from this history is that this is a question that's actually older than science itself. So it's actually interesting to recall that one of Aristotle's objections to the kind of totalitarianism that Plato espoused in the Republic was that even under this scheme, people are actually inevitably going to recognize that there are certain people they resemble more than others. And in this way, the family ties that Plato was trying to abolish will start to creep back in and lead to a destructive tension with the official attempts to stamp them out. And surely, the matter of heredity was something pondered by at least some people even before the time of the ancient Greeks. Nowadays, we would probably put the question something like this. How is it that biological endowment interacts with experience and culture to produce behavior? So here's another scientific motivation, focusing on qualitative properties of the human mind as opposed to individual differences. So there's a school of thought in cognitive science that used to be called nativism. So the basic idea of nativism is that the human mind possesses certain capacities, some of them uniquely human, others perhaps shared with other animals. And these are called by different theorists modules or faculties or core systems. Perhaps the most famous example of these is Noam Chomsky's innate universal grammar, which has been called by others the language acquisition device or the language instinct. Now, a relevant question, which used to be entirely neglected but seems to be attracting more thought nowadays, is whether it's possible for the actual genome to encode capacities such as this, and if so, whether empirical genetic evidence can be brought to bear on these hypotheses. and give this discussion less of the armchair philosophical character that it has traditionally assumed. So finally, behavioral genetics is important because of its connection to the study of evolution, which can be defined as the changes over time in the frequencies of alleles, that is, classes of genetic types, such as the famous GCTNA. Now, the subfield of psychology called evolutionary psychology treats human nature, by which is meant not only cognitive capacities such as language, but also, for example, our sexual and moral instincts, and also social behaviors such as religion. So evolutionary psychology treats these things as products of biological evolution. And it seems to me that this approach can only benefit from the deciphering of natural selection and other evolutionary forces in modern genetic data. So I've laid out these promising connections between behavioral genetics and other parts of psychology, and indeed science at large. Despite these connections, however, behavioral genetics has traditionally been somewhat of an isolated and insular field. We can see this in the fact that the vast majority of psychology departments do not have a behavioral genetics program. So up in Minnesota, we're sort of oddballs in this way. So maybe that's okay. That just means more glory for us. But I think that the neglect is actually worrisome to the extent that it goes in both directions, such as when behavioral geneticists, almost with pride, admit that they don't really know much about the brain or anything else. Okay, so given this historical inertia, whatever its causes, that could be a whole lecture in its own right, we have to ask ourselves the question, is it actually worthwhile to, I suppose, have events like this and try to make genetics a more integrated part of conceptually neighboring intellectual disciplines? So I'm now going to show you a video of some twins separated at birth who participated in a study conducted in my department by my predecessor, Tom Bouchard. This study is called the Minnesota Study of Twins Reared Apart, or MISTRA for short. Now, my own PhD advisor, Steve Pinker, he was in MIT at the time, he was interested in questions of evolution and human cognition at the time that the paper reporting the original MISTRA results came out in Science back in 1990. He had never heard of behavioral genetics at the time, but when he learned of these results, he was so blown away that he's permanently incorporated behavioral genetics into his own thinking. And in fact, he's a co-author of one of our papers. So let's watch this video. The psychologist that you'll see in it is Nancy Siegel, who was a postdoc in Minnesota at the time that MISTRA was carried out. She's now a professor here at Cal State Fullerton. All right, so let's take a look.
All this made us wonder, what makes us who we are? Is it how we grew up? Or is DNA our destiny? Hi, Tamara. Researcher Nancy Siegel has spent a lifetime trying to answer these questions. She has interviewed more than 50 pairs of identical twins who were separated at birth.
Identical twins raised apart are exceedingly rare. They're very, very valuable cases for scientists. We're very interested.
She wants to know how Tamara and Adriana's similarities compare with other identical twins separated at birth. What can they teach us about why we behave the way we do? Why we pick certain jobs, certain friends, even certain shampoo?
To the extent that identical twins are more alike in these things, we can say with a fairly strong degree of confidence that yes, genes do play a role.
48 Hours found 4 sets of these scientific gems and brought them to New York.
All 4 of you, 4 pair, have had a similar experience. You were identical twins, separated at birth, adopted and raised by different kinds of families, and then came together as adults.
Identical twins Tom Patterson and Steve Tazumi met just 4 years ago. Tom is from rural Kansas. Steve lives in urban Philadelphia. Their upbringings couldn't have been more different.
I was a Christian and he was raised a Buddhist. His dad was a pharmacist and my dad and mom were janitors.
Still, Tom and Steve chose the same careers.
It's phenomenal is that he owned a bodybuilding gym and I owned a bodybuilding gym. So we're both definitely 100% into fitness.
Yes.
Daphne Goodship and Barbara Herbert first met when they were 40. They too discovered remarkably similar life experiences.
We discovered we'd had a miscarriage the same year, followed by 2 boys and a girl in that order. We've even had cooked the same meal from the same recipe book.
On the same day?
Yes.
Without knowing?
We've done it a few times, but that one we can remember clearly.
And you were raised Jewish, and you were raised...
I was raised Catholic and now I'm Christian.
Debbie Melman and Sharon Posette were brought up in different faiths with different traditions,
Still that boy.
but their habits are incredibly similar.
When you get excited,
We
you cross
cross
your...
our eyes.
You do this?
Yes.
Oh, my God.
And our dads would have said the same, like, if you roll your eyes at me, young lady.
Of all these twins, Roger Brooks and Tony Malassi have the most dramatically different backgrounds.
Well, I was in an orphanage home the first 5 years.
While Roger bounced from home to home, Tony was adopted by a loving family.
I had a very secure, good childhood, a lot of love in the family.
Do you see a big difference because he didn't have that early stability?
No, I don't. I didn't live in a traumatic environment. I lived in a good environment. You had rickets? No, he did. His shoes were tight. He doesn't realize how tough he had it.
Having gone through all of those difficult times, Roger is still an upbeat, positive person.
Still Tony.
Yes. I think it speaks to the genetic predisposition to just simply have a sunny, upbeat, happy personality.
Research shows that identical twins raised apart are more alike than they are different, and they're surprisingly more alike than the brothers and sisters they are raised with. We asked Nancy Siegel to design an experiment to show how genes play a role in our everyday choices.
We were interested in their choice of places to go on vacation and what kind of foods they might choose given an array of choices. How they moved and gestured.
We'd like you to stand on one foot.
Barbara and Daphne are called the Giggle Twins, and I think you can understand why.
And they can't see each other. This is a
No, that's a
partition.
partition. They can't see each other.
How are you most comfortable?
Oh, look at that. Oh, now,
They're doing this
they're
without
not
seeing
sure
each
which is
other.
more comfortable,
But
and
they keep doing it exactly.
they're not certain which is more comfortable. Now we've asked them to dance. We want to see how their bodies move, and they show a lot of similarities. See how they're moving?
Look at that. Oh, look at that. Oh, my.
See, here they can't see each other, but they're moving the same way.
I just want everybody to pause. You're sitting there like this. Get this picture. And you're sitting there like that. Your legs are crossed. Can you see this? I can't see what you guys are doing back there. Your hands are the same? On the knees, both of you?
They're the same
Oh,
thing.
my God.
What do you make of the weird coincidences? I wouldn't call them weird coincidences because I think they're scientific phenomena. If you think that all of us are more comfortable in certain ways because of the ways our bodies are made, and if all of us had identical twins, they'd be sitting exactly as we are.
You're saying that somebody would cross their hand this way because their body is made in such a way that this just is the most comfortable way to
Exactly.
sit.
Siegel believes that's why Adriana and Tamara dance the way they do.
One could say, well, their mothers gave them dancing lessons. But the question is, why did their mothers give them dancing lessons? Because the girls were agile, enjoyed it, did well at it.
Adriana and Tamara, like these separated at birth twins, believe they are in fact more identical because they were not raised together.
You don't have that baggage of, oh, mom, like, I mean, I'm sure or like, you stole my boyfriend, or you studied and you got a 92, and I got an 88, or something, you know.
Because they never had to compete with each other, they believe they were freer to express their genetic potential.
You are what you are.
You're genetically
Yeah, I think
packaged.
so, yeah.
So does it...
All right. Still get the chills every time I see that. So actually, interesting thing, I actually first learned about MISTRA, the Minnesota Study of Twins Reared Apart, from my future PhD advisor at the time by reading his book, The Blank Slate. Now it's difficult to say for sure that reading this book or meeting that person or having this conversation actually changed your life. I mean, as we just saw, the puppet masters pulling on your strings are not necessarily what they seem to be. But I'm willing to go out on a limb and say that Steve, Tom, Nancy, my current colleague Matt McGew, everyone who worked on MISTRA really did change my life. After reading about this, I realized that what I was doing at the time was not for me, and now here I am standing in front of you instead of slaving away for a big law firm in New York or Washington, D.C. or whatever. Okay, so this is actually why many of us find this so worthwhile. When there's something like this out there, waiting to be understood at so many levels. Then as a scientist, you just feel very lucky that you know about it and to be hot on the trail. So, so basically from the results of MISTRA and many other behavioral genetic studies, we can say that the genome appears to affect not only very coarse phenotypes such as general intelligence, but also very idiosyncratic and minute aspects of personal identity. And traditional and modern DNA-based genetic studies are basically trying to get to the bottom of this striking phenomenon. I do want to add a caveat, however. So these remarkable similarities that are observed between the identical twins reared apart, such as both owning bodybuilding gyms or whatever, anecdotally at least, these are virtually never observed in studies of other kinships, not, for example, in dizygotic twins reared apart. And this is almost certainly because these very specific particular outcomes are the result of what is called epistasis. This is just the genetic jargon for nonlinear interactions between different sites in the genome. Now, epistasis is going to come back in the story later that I'm going to be telling you today. And I think that we can learn some things about it. But it seems to me, just at an intuitive level, that some of the deepest secrets of this phenomenon, like the things that we just saw, may continue to elude us for quite some time yet. Okay, so let me talk about something that I think that we can make some progress on. So I'm going to be talking about a class of problems that I think genome-wide association study data, GWAS data for short, may be able to help us make progress on. So it turns out that sexuality and sex differences are very popular topics in psychology. In fact, evolutionary psychology has even been called derisively the psychology of mating. Somebody mentioned to me yesterday that they were at the HBES meeting. I've heard that that's the meeting that's known as the hookup meeting in psychology. So certainly this is something that attracts a lot of interest. But these surface phenomena actually overlie a deeper and more fundamental issue. So before we ask questions such as, well, why is it that girls tend to get better grades in school, or why the vast majority of prisoners are male, and so on, we can ask, why is it that we even have two distinct sexes at all? Like, you can imagine that when it comes time to reproduce, you would just pluck anyone at, potentially you can mate with anyone in the population. We could imagine that, but the reality is that your choice of partners is restricted to a special subset that is roughly 50% of the population. So why are things like that? Furthermore, the complex process of sex itself, featuring these male and female specializations, doesn't actually seem to be logically necessary. So why not actually reproduce asexually? That is, once you've had tenure and so on, it's time to move on to other things. You could just butt off a little copy of yourself at that point. So once you start to ponder this question, it can have a way of intruding into all of your thoughts about sex. Well, I don't know about your thoughts. Maybe it's just my thoughts. I mean, the way the biologist William Hamilton put it, this is the formulator of the so-called Hamilton's rule in the study of altruism. Sex is an intellectual problem for some of us, but a personal problem for us all. So that's how he described this double way of thinking. And just to drive home the point, here's the picture from my notes on genetics about how sexual reproduction works, where the father and the mother contribute a random half genome, that's a gamete, and then the gametes come together to make a member of the new generation. This features all kinds of complicated machinery for replicating the DNA and properly segregating it to the two daughter cells and so on. Whereas it seems like life would be much simpler if we could just do things like this. Okay, not only does it just seem simpler, if you think about it just a little bit more, there seem to be these obvious disadvantages that reside in sexual reproduction. So let's compare a hypothetical sexual population to an asexual one that's very similar to it. What does very similar mean? That means that these two species are essentially competing for the same niche in the economy of nature, as Darwin put it. So in this asexual population, so let's imagine that the only limitation on how much an individual can reproduce is their own capacity, their own resources, so on. There's no external density limitation imposed by the environment. So in the asexual population, every member is female, And if each such female has 2 children, then the asexual population doubles in size each generation. However, in the contrasting sexual population, since only half of the population is able to give birth, then the same birth rate, 2 children per female, keeps the sexual population at a constant size. And if you want to think of it more realistically as there being some kind of external limitation on the total size of the population, we could think that this niche in nature for which these 2 populations are competing, if it has a finite number of slots, then the asexual population should be able to fill those slots way more quickly than the sexual population can. And either way, it should be the case that the asexual population should quickly swamp the sexual one. This thing in the literature is sometimes called the cost of males. Like, why is it that we have all these worthless males around? So it's actually greater than implied by the preceding argument because, as should be clear, mating can be dangerous, time-consuming, energy-draining. Furthermore, the sexual act can transmit diseases to the partner or the resulting offspring. And also, sexual reproduction also causes what's called sexual selection, which can lead to traits that actually impair survival, such as the peacock's tail, perhaps being the most famous example. Perhaps from the point of view of some members of the population, the greatest cost of the sexual system, so to speak, is actually the possibility of failing to reproduce at all. So this actually happens in some species of walrus, for example. It will turn out that a small fraction of the males father the entirety of the next generation. And so if you're one of the unlucky males who's not big enough, not fierce enough, or whatever, it seems like you might ask yourself the question, why not have at least the option of reproducing asexually? Like, whatever the benefits of reproducing sexually are, if you're not able to secure them, why not at least have the option of reproducing asexually? And actually, there are some species where you can't switch back and forth, but in a lot of cases, obviously, you can't do that. So apparently, in those latter cases, natural selection favors the strategy of continuing to try, no matter how hopeless it seems. Okay, so here's a way to see that sexuality, paradoxically, after going through all this, apparently, is actually what natural selection seems to want in vertebrate species, at least. So let's presume that asexuality was the initial state, because today, we see that the species that are asexual are typically unicellular organisms that are probably not too different from our common ancestor. Now, it is true that you can see little twigs in the tree of life here and there, vertebrate species that do reproduce asexually, such as, for example, this species of lizard that lives in Texas. So from those little twigs that pop up now and then, it seems reasonable to suppose that a switch to asexuality is a possibility. It's not like it's like a one-way path where going in reverse is like physiologically impossible or something. It seems that, that the switch back to asexuality is an option if it is not disfavored by natural selection. But the thing is, when you look at where asexuality exists on the tree of life, these are indeed twigs. That is, it turns out that most asexual animals, we can tell from phylogenetic analysis, are recently descended from a sexual precursor. Long established asexual animal species, that is, animals that have switched to asexuality, have been doing it for millions and tens of millions of years, these are virtually non-existent. And we can infer from that that animal species that become asexual will either revert to sexuality or become extinct. So therefore, it must be the case that sex confers an overwhelming advantage. But kind of the problem is, if it's so overwhelming, I mean, why isn't it obvious what it is? So that's the problem of sex, the intellectual problem, at least. There was a question?
You're saying it's specifically in animals, right?
Yep.
Like fungi and other species do have more long-established asexual reproduction?
There's a species called the bee, I forget what the bee stands for. The genus starts with B, rotifer. And they have reproduced asexually, it's inferred, for 40 million years. But there's sort of the exception that proves the rule, and there's armies of people studying them just to figure out what is going on with them, yeah. Yeah.
So I thought the question was about like fungi and things that reproduce asexually that aren't animals, and it seems like there's a lot of those.
Yeah, so part of the problem is to figure out what is different about those from things that do reproduce sexually. because it seems to be a consensus that I'll explain this a little bit later. It seems that in species that are extremely large, that is, their population size is huge, basically all, maybe this will make sense later, but basically you can imagine that the perfect type, the genetically perfect type, can just arise by mutation because the population size is so huge and you have so many chances at it. And so therefore, in such a population, you do not need sex for the reasons that I'll go into later. Is that satisfactory for now? Yes, that's an important thing. This was actually something Hamilton emphasized, that the correct theory has to explain why sexuality is mandatory for some, facultative or optional for others, and is not really used by yet other species of animals, or bacteria and so on. Okay. So there are qualitatively different families of hypotheses that have been advanced to try to solve this problem. I'm going to talk about just one of these, which the theorists now all seem to agree has at least some truth to it, and actually which, well, there's a secondary importance, but this is actually something that GWAS data might be able to help us confirm. But to understand this particular class of hypotheses, we need to define a particular concept that was in my notes on genetics. This is called linkage disequilibrium, or LD for short. So basically what this means, suppose that in the population at large, the count of fitness-enhancing alleles, that is the gene variant that increases your ability to survive and reproduce, as opposed to the other variant which decreases it. Suppose that the count of the fitness-enhancing allele at one site is negatively correlated with the count at another site. So this is just a negative population correlation. If that's what we observe, we're going to say that these two sites are in negative LD. So this is a figure from one of our papers. It's basically a color-coded correlation matrix of chromosome 22. Interesting thing is that it is almost diagonal. That is, large correlations are usually only spatially close by, whereas two sites that are very far away are usually not in any detectable LD. The theories that I'm going to be talking about later about why sex is advantageous require a long-distance LD. But the thing is, that long-distance LD between any parasites could be still very small. That is undetectable in something like this. And it could still be the case that the theory still goes forward. It's just something that we're going to need larger sample sizes to measure whether there is this very, very low levels of LD that might be required for the theory to work. Okay, so for now, let's just assume that negative LD, as I've defined it, between different fitness-affecting sites in the genome is the rule. Yeah, there's a question?
Yeah, for the last thing you said,
yep.
You mean distance as in actual distance or something like centimeters?
Either. So centimorgan just means frequency of recombination events between the 2 points. Naturally, as that goes up, any initial LD there is between those 2 points will decay. And genetic distance is correlated with physical distance. So we're talking this loosely, either way. Okay. All right. So without explaining why this might be the case, let's just assume for now that distance sites in the genome that affect fitness will tend to show negative LD. Okay? I'll explain why that might arise later. Okay. So negative LD means an excess of plus minus and minus plus haplotypes. Excess with respect to the product rule that leads to the expectation you would have for these two events being totally independent. So if you are plus minus at these two fitness affecting sites and you happen to reproduce asexually, then since your clonal offspring is just essentially like an identical twin of yourself except born at a later time, then your clonal offspring will also inevitably be plus minus at those two sites. Okay, that's just how asexual reproduction works. But if you're a sexual creature, then the situation is quite different. So by recombining the genetic contributions of your own sexual parents, you can pass on plus gamete to your offspring even if you don't have such a gamete yourself. That is, by breaking off a piece of your dad's contribution, connecting it to a piece of your mom's contribution, even though you don't actually have a plus haplotype, in theory you can pass on a plus haplotype to your own offspring when it comes time to be a parent yourself. And so here's another visual way of illustrating how this process affects the population statistics. Suppose that for whatever reason, every haplotype in the parental generation is either plus minus or minus plus. If the effects of these two sites on fitness are equal, then the heritability of fitness, that is the percentage of the population variation in fitness that's due to genetic variation, then the heritability of fitness in this population is 0 because there are in fact no genetic differences of fitness. Okay? That is, if you're looking at the distribution of different genetic values of fitness, there's actually no distribution. There's just a pile of probability on one value. However, once sex and recombination have done their work from such a parental generation, you can have an offspring. You can now have in the next generation offspring representing the two haplotypes that were previously missing. So in other words, in the offspring, plus and minus are not as disproportionately paired. Those two extreme haplotypes, plus and minus, now occur at the frequencies that are maybe not quite there yet, but closer to those that are predicted by the product rule for independent events. Now, what is so good about that? Like, why is the population in some sense better off after that has happened? Well, intuitively, this is because natural selection can now has an easier time increasing the frequencies of the plus alleles at the two sites. Now, before, natural selection was in fact impossible because everyone was genetically equivalent. If one person survived better and reproduced more than someone else, this was totally irrelevant to the future genetic composition of the population because the person who reproduces more is actually genetically exactly the same as the other person. But after sex and recombination, natural selection is possible. The plus alleles can now increase in frequency because now there are genetic differences in the population. And it now matters whether one person survives and reproduces more effectively than another. Okay. This insight can be expressed more formally in terms of what's called the breeder's equation, which I've displayed here. So the R stands for the evolutionary increase in the mean phenotype. That is how much the average increases from one generation to the next as a result of natural selection. This being an average of whatever phenotype you're interested in, which if you're a livestock geneticist might be milk yield, or it might be bristle number in fruit flies if you're a Drosophila geneticist. The trait that's represented here can also be fitness itself, in which that case the breeder's equation becomes Fisher's fundamental theorem of natural selection. But let me put that aside for now. So we can see that the increase in the average of the trait is a product of two factors. The first is the heritability of the trait, which I've just briefly mentioned. So again, this is the fraction of the total trait variation that's due to genetic differences. The second factor is the mean trait difference within the parental generation between those who survive and reproduce and those who do not. Although when we're talking about Fisher's fundamental theorem and the trait is fitness itself, it happens that this just becomes the total variance in fitness, which leads to cancellations, and so this becomes the total increase in fitness becomes the additive genetic variance in fitness, but whatever, I guess that's for aficionados. Now, recall that if we start with negative LD, and then sex and recombination does its work to decrease the negative LD. What this does is that, as we saw, it increases the variance in the genetic values of fitness. And so therefore, it increases the numerator, well, increases the denominator as well, but the net effect is for the heritability to go up. And so with this factor being larger, it turns out that the sexual population, its ability to adapt to its environment, its average ability to survive and reproduce, will increase more rapidly. And in fact, if there is sufficient genetic variation in fitness due to a large number of fitness-affecting mutations entering the population each generation, then it turns out that the advantage enjoyed by a sexual population can be enough to actually overcome the twofold cost of males. So that's something that can explain why sex is helpful. It helps address, it will help a population if it's sort of habitually in negative LD between the different sites in the genome that can affect fitness. But now let's return to this initial assumption that that's the way things are. Why might actually negative LD arise in the first place? So there are actually several possible, some of the explanations that have been offered for this. Some of them are closely related. Here we'll examine only one of these possible explanations, and I'll introduce it by way of what I often do in my class, which is I'll often pause the lecture to ask the class questions, and then they vote using their clickers on what they think is the correct answer. So here's the question that I use to explain this particular concept, which actually surprisingly arises in many different contexts other than this particular one. But so here's how the question goes. The correlation between SAT verbal and math scores in the general population is, this is actually true, it's roughly 0.7. Scores on each section range from 200 to 800. The mean is 500. The standard deviation is 100 when we're talking about one of the given sections. Well, this was a while ago. They changed it since then, the scaling. But all right, now suppose that we examine a selected subpopulation where all individuals obtained a combined verbal and math score of 1560 or higher. What will be the sign of the verbal math correlation in this special club, this specially selected subgroup? Okay, now I've asked this question, I think, 3 years, and every single year, exactly 0 students answer this question correctly. It's pretty dispiriting when there's like 100 kids out there and 0 out of 100 answer it correctly, even after a chance to discuss it. But I guess that's what that's telling me is I should stop asking this question. But anyway, the answer is actually that the correlation becomes moderately negative, and this is the explanation why. So if we know that a person in the special club, the specially selected subgroup, scored 760 on the SAT verbal, then we know immediately that the person must have scored 800 on math, or else they would never have been in this special club for us to be able to observe them. Okay. Likewise, knowing that a person scored 760 on the other section, math, means that they must have scored 800 on verbal. So in this way, within this specially selected subpopulation, relatively higher scores on one subtest go with relatively lower scores on the other one. Questions? Let's start with Dalton.
Next year, can you take, you
Yeah.
have the picture of the scatter plot
Yeah.
for the full distribution? I think that might be misleading your students.
Yeah.
Take the picture out and see if anyone gets it.
So instead of, so don't have this and
Yeah,
see
don't
it.
have that.
Yeah, maybe. I thought you were going to say that I should draw like lines or something to isolate this.
You see it, and
Yeah.
don't show them the subgroup, but
Yeah.
just don't show them anything.
I mean, you could show that, just clear on the part that you're talking about to see if you, in theory, if you could zoom in on that part of the distribution.
Okay. Well, I'll consider it. Yeah. Okay. There was another question somewhere.
What you were talking about, like collider bias earlier.
Yes, this is intimately related to collider bias. Yes, it's just an example of collider bias.
Zero of your students get this. I think you should draw the line. So you make it what you think is going to be a super easy question, and
Yeah.
then see how many get it. If you draw the downward sloping line and then it cuts off the thing,
Yeah.
maybe they can see it with their eyes, and that if they don't believe their eyes, then
It's like.
no,
Yeah.
it's a straight line. It's a downward sloping line.
Well, then making 7.
You mean just through the like this?
Yeah.
Through the.
From 780 to 780.
Yeah. Okay. Yeah. Okay. That's. I mean, I'll give it a try. I'll definitely give that a try. Yeah. Okay. Let's see. Where am I? Okay. So this is interesting. So selection is understood broadly can not only increase the frequencies of certain objects, it can also change patterns of relationships between objects. In this particular case, it has turned a positive correlation into a negative one. I once explained this to someone, and they said, "James, do you want to try to destroy all empirical research? Why are you bringing this up?" But anyway, okay. So here's the thing. This is not something that's just about SAT scores or whatever. This is actually a principle that also applies to natural selection. Suppose that selection works in the following way, that individuals who do not carry enough of the plus alleles, so to speak, simply die or reach the end of their lifespan without reproducing. So among the survivors who do reproduce, what can happen is that the count of plus at distinct pairs of fitness-affecting sites can become negatively correlated. That is, if a survivor has a count of 0 at one site, it becomes slightly more likely that the survivor has a count of 1 or 2 at another. So here's a visualization of this working. So all these individuals who are loaded with too many minus alleles are eliminated. Then where are the surviving minus alleles, so to speak, going to be found? Well, naturally, from this point of view, they're going to be found sort of hiding within other genomes that contain plus alleles to sort of compensate. And in the meantime, there are new mutations, most of which are harmful if they have any effect at all. Okay, so I personally like this hypothesis because it ties into other things such as collider bias and so on. But it turns out that this explanation of why there might be negative LD and thus leading to a benefit of sex, this is not any longer as widely accepted as it once was. The reason for that is that it only works if epistasis, that is nonlinear interactions between different parts of the genome, only if epistasis for fitness has a certain functional form. And recently, people have expressed doubts based on empirical studies or whatever that whether epistasis generally in nature for a wide variety of species actually does have that functional form. However, there's a closely related kind of explanation that I don't really want to go into today. It's sort of more complicated. This invokes both the effects of selection and finite population size. So earlier I was talking about how the size of the population, large-bodied animals like us typically having much smaller populations than bacteria and so on. When you combine effects of finite population size with selection, it turns out that you can show theoretically that this will also lead to negative LD. And then population is once again in a place where sex can actually be beneficial.
Why does your Simpson's paradox explanation require any epistasis just purely on an independent additive basis, I thought?
So in the examples I had, yes, if different sites in the genome just add up perfectly, then yes, cutting off people who are not fit enough will lead to negative LD among the survivors. But it turns out, for example, if there's what is called antagonistic epistasis, which is that there's sort of a saturation effect such that additional bad alleles don't actually decrease your fitness all that much, then selection will actually cause positive LD. And then if there's positive LD, sex will actually decrease the heritability of fitness and then leave the population in some sense worse off. So basically, in order for selection to cause LD, there has to be the opposite of that pattern, which is called, what do they call it? Synergistic epistasis, where additional mutations hurt your fitness even more. So anyway, this is an issue that continues to be debated. There was just a paper in Science a few months ago reporting that there's negative LD between loss of function protein coding mutations in human beings and Drosophila. And then they argued based on simulations of human demography that finite population size effects are not enough to produce this. And then they argued by inference, therefore, it must be the case that synergistic epistasis is the rule. But anyway, yeah, this is something that it goes on. Okay, so finite population size. This is often called the Hill-Robertson effect, named after the quantitative geneticist William Hill and I believe it was his advisor at the time, Alan Robertson. So for those of you who are aficionados, you might be interested to know that William Hill is actually the PhD advisor of both Peter Vischer and Naomi Ray. So a little bit of history there. Okay, so speaking of this being sort of a very active field in light of all the new data that we're able to accumulate now at the DNA level. So there was a paper in Nature that came out roughly 15 months ago. This was a study of experimental evolution in, I forget the species now, but it's like C. elegans or one of those. So basically, this is a kind of experiment where you can actually follow evolution over time. You can follow a population as it evolves in its petri dish and it samples the time. You can take some of them out and sequence their genomes and look at the genetic changes that are going on. And this study actually found very striking support for these Hill-Robertson, Fisher-Muller class of hypotheses. They basically found that yes, before the population becomes sexual and starts evolving, you do see an excess of plus minus and minus plus. And they also have these tricks that will prevent the species from switching to sexuality, and they can see how the fitness deteriorates in that population as all these negative mutations interfere with the fixation of plus mutations. In contrast, you can observe in the sexually reproducing lines that that this interference is broken up by recombination, and then the positive mutations go up in frequency, the negative mutations go down, and these things dominate their little petri dish much more effectively than their unfortunate rival populations in the other petri dishes. Okay, so this is a very exciting result, but I think a question that naturally follows is that can we generalize this result to natural populations living out in the wild as opposed to things that live in petri dishes? I think the answer to that is yes. So my colleagues and I, have, including some people in this room, have recently published a GWAS of number of children. This came out in Nature Genetics last December, although stunningly it's only been cited 4 times. But so basically, this was a study of a number of children that use a sample size of roughly 330,000 people. Actually, interestingly, it only found 3 hits for number of children. So this is sort of the most refractory phenotype to GWAS that's been studied so far in terms of how many hits do you get for a given sample size. But in retrospect, that's not so surprising because if you think about it, essentially anything could affect your fitness. So a bit hard of a row to hoe ahead. But anyway, this work, basically, if you don't care about identifying individual SNPs that are associated with fertility or fitness, you just care about certain aggregate quantities. This work might allow us to test whether SNPs that are associated with fitness in a natural population, such as human beings. What I always say is that human beings are a great population to study. Imagine trying to get 1 million butterflies to give you their DNA and all their little phenotypes. Whereas in Homo sapiens, you can just herd them all, and it's just. But anyway, and so here's what I sort of imagine. So on the x-axis, we have, for example, products of GWAS regression coefficients averaged over many, many, many SNPs. On the y-axis, we might have the linkage disequilibrium polarized with respect to the fitness increasing allele at both sides. And so what I would hope to see, by the way, if there's anyone who's very interested in this and they're willing to devote their heart and soul to this, for a short time, a limited time, this is not like selling your soul to the devil, please come talk to me because I've been interested in this for a long time and I've just not had the resources to sort of get going with it. But anyway, if we could do this with like the UK Biobank or whatever, and if we saw a pattern like this, where if the 2 alleles that tend to increase fitness tend to be in negative LD, and the strength of this relationship increases with the effects at both of the sites that you're examining, then this would be very powerful evidence that, yes, even in natural populations, this kind of selection, perhaps combined with finite population size, leading to negative LD, which eventually will sort of crush an asexual population, can be undone by sex. And this is why that this is something that's so great and wonderful. So here's a caveat. So if we did this study and we actually found positive LD or even maybe no LD to the extent that we can determine. Oh, one thing I want to point out. I don't know if these numbers would be accurate. I just sort of made them up. But the point that I would like to emphasize is yes, whatever the real numbers would be, they would be in fact extremely small. But it has been my experience when plotting these aggregate quantities, such as allele frequencies versus average betas and so on, that once the GWAS has reached a sample size in the hundreds of thousands, if you aggregate over enough SNPs, you see these extremely regular patterns despite the scales of the two. For example, the y-axis being extremely small. So, So this would be like the correlation between this, the fitness increasing alleles. Like yeah, R or D or something like that. Okay, so here's the thing. If we observed positive LD, this would not necessarily rule out this class of hypotheses, because it could be that positive LD is caused by assortative mating. And so it could be that sex plus assortative mating not only moves the equilibrium to something slightly less negative, but moves it all the way over to the other side and makes it positive. So in some sense, this is not a powerful empirical test that we can do. However, the flip side is that, if the finding is that, as a rule, fitness affecting sites in the human genome show negative LD, then there's only one possible interpretation of that, which is that there is some kind of selection plus finite population size effect that does in fact lead to this negative LD, and therefore we adapt to natural by means of natural selection more effectively as a result of our sexual nature. Okay, so I've been talking about why animals like us reproduce sexually instead of clonally. But let's return to one of the original questions that I posed, which is why have two sexes, each apparently specialized for a different role in reproduction? Why not just be able to reproduce with, even if you have to reproduce sexually and thus find a partner, why can't that partner be any member of your species instead of a member belonging to this special subset making up roughly half of the species? So we can imagine such a state, no specialized sexes, no male, no female. So in this scenario, even though reproduction is sexual, you can mate with potentially any member of your species. And there have actually been science fiction stories about this by like Isaac Asimov and people like that. So the state that I've just described, sexual reproduction but no specialized sexes, this is called isogamy. In contrast, if each partner must be of a distinct specialized sex, then the species is said to practice anisogamy. Okay, so how might we think about why anisogamy is much, much more common in nature than isogamy? Although you still find isogamy here and there, like for example in this species of green algae. Well, let's imagine shortly after our lineage started to reproduce sexually. So imagine at that point in time, maybe 800 million years ago or something like that, in a primordial marine ancestor that lacks specialized sexes. Okay. So back at that time, what has to happen is that each hopeful parent just sprays gametes into the ocean. And with luck, your gamete will unite with a gamete that was sprayed by some other member of your species. They'll form a zygote, which will then develop, hopefully, into an adult. Okay, so let's make two assumptions, very plausible assumptions, I think, about how this process works. The first assumption, let's say that each parent has a fixed budget for reproduction. So this means that it can make many small gametes, or a few, or even one, like really big one. Okay? So there's some trade-off between size and how many. Assumption two, bigger gametes lead to a bigger zygote, and bigger zygotes in turn are more likely to survive and reproduce. Okay? So the idea is like more yolk for the little chick or whatever, the better start it has in life. Okay, so let's imagine this is a visualization of that primordial state. So you have all these parents who have basically the colors represent different gamete sizes. The blue parents go for the big end of the trade-off. They produce a few large gametes. The yellow parents go for somewhere in the middle, a few intermediate-sized gametes, whereas the red parents just produce a lot of gametes, and they're very, very small. Okay, so now the gametes scramble around in the ocean trying to find partners, and they pair off in the following way. Now suppose that only the zygotes that are formed from large, that is blue gametes, survive. That is, you need to have a contribution from a gamete of that size in order to cross the, say there's a sharp threshold for whether you survive. Okay, so then the other zygotes are not big enough, they're all killed off. Oh, ho, 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 look at what we have here. An epochal event in the history of life, not something new appearing under the sun. Now, in the next generation, the spraying of gametes occurs again. And look what happens in our warm little pond. Now, there are only big gametes and small ones. Okay? So as a result of this natural selection, organisms producing middle-sized gametes ultimately have no descendants. And eventually, in later generations, every parent is now one of two types at the extremes of the original trade-off. Every parent is either a producer of large gametes or small ones. Okay, this process of natural selection can continue even further, resulting in extreme specialization. So nurturers, I'm now using Richard Dawkins's terminology. It's sometimes helpful pedagogically to avoid using the terms male and female until later. But so at this initial phase, let's use this Dawkinsian terminology. So nurturers produce a few large gametes full of nutrition, giving the zygote a good start in life. However, the producers, the other type, the exploiters, they produce numerous small and mobile gametes, bringing little nutrition to any given union, and instead trying to form as many unions as possible. And so, and there follows our ejection from Eden, I suppose. All right, so there's a problem that arises in this theory of why we have two sexes, which I actually think that GWAS data can help. In fact, GWAS data has already helped. So notice that we have a problem here in that natural selection wants the nurturers on the one hand, the exploiters on the other, to have different phenotypes, that is, to have different average characteristics. But a gene that is a little like token of hereditary material as it's copied and transmitted across the generations will actually spend 50% of its time in the body of an exploiter and the other half of its time in the body of a nurturer. And so basically, how can sex differences be genetically caused if a parent of a given type transmits the same genes to its offspring of both types, okay? So suppose you're like, like a Genghis Khan or somebody like that. You're very, very good at the exploitative strategy and you have tons of offspring. But the thing is, well, this is actually a question. Why is the sex ratio 50-50? There's an explanation for that as well, but I won't get into that today. But just assuming that the sex ratio has to be 50-50, half of your offspring are going to be exploiters, half of your offspring have to be nurturers. And so the thing is, even if you're genetically like supercharged to be like a great exploiter, that's probably going to be ruinous for your children who are nurturers because the optimal strategies for the two types are so different. Well, the solution to this problem is that it appears that, well, initially you could just posit, well, let's just imagine that there are certain genetic sites that have a phenotypic effect in only one sex, or maybe opposite effects in the two sexes or something like that. Well, anyway, some kind of dependence of the effect on the sex of the body in which it finds itself. And it turns out that this is not something you can just posit to sort of make this theory work. There are actual examples of sex-dependent effects, for example, from GWAS of waist-to-hip ratio. It turns out that, so waist-to-hip ratio is something that's close to 1 in men, but in women, on average, it's smaller than 1. The reason why for that is that the hips have expanded to accommodate the larger heads that humans tend to have. So this is an average difference between males and females, and it turns out that many of the GWAS hits for waist-to-hip ratio essentially have effects in women only. So suppose that you're a woman and your genotypes at these sex-limited sites cause you to have a below-average waist-to-hip ratio, the better to have big-headed babies. Well then, 2 million years ago or whenever this was, your daughters are more likely to have below-average waist-to-hip ratio as well. However, but your sons are not, okay? And in fact, if you estimate the genetic correlation between males and females for waist-to-hip ratio, it's anomalously low. It's like around 0.5. So yay, we have the Dux ex machina that will allow this theory to work. Actually, to be less flippant about it, probably the mechanism by which sex-dependent effects can occur is that the cellular environment where the gene is expressed, it probably will be different depending on messages that are received from sex hormones that bind to receptors on the surface of the cell. And once that difference is there, the internal environment can detect it, act on it, and therefore produce different phenotypes depending on the signals that it's transduced. Okay, so once certain causal sites can have effects in one sex only, Natural selection can optimize the two sexes in different ways. And the conclusion is that natural selection can indeed make nurturers and exploiters very different, despite the fact that individuals inherit genes from a parent of each type. Okay, in conclusion, GWAS and similar types of studies of genome function can fill in many important details about the answers to this family of questions that I've been talking about. For example, what fraction of the genome is functional? That is, what fraction of the genome, there's a certain fraction of the genome where if mutations occur there, there'll be no phenotypic effect whatsoever. And so what are the respective proportions? How many deleterious de novo mutations does the average person carry? This is a crucial parameter for making these selection plus finite population size arguments work. What is the heritability of fitness, and how much is it going to be modified if we did not have so much recombination, and so on? What is the functional form of epistasis? How often do we find sex-dependent effects of common variants or de novo mutations? I mean, once we have answers to all these questions, it would seem that we would have this pinned down at long last. Okay, so I hope I've communicated the fact that this is a very exciting time to be interested in these questions. It's actually pretty amazing to think that we may soon find a satisfactory answer to the enigma behind this most profound source of joy and misery in the human condition. Okay, how much time do I have left? Should I go through my summary slides, or there's a question.
Well, can you just say a little bit more about the finite population size?
Yeah.
And how that works. I assume it's complicated, but I want to hear a little more.
Okay, so the way Fisher put it initially back in 1930, he said, like, in a finite population, suppose there's two sites, and then somebody in the population has a good mutation at site 1, say. But then suppose that someone else in the population has a good mutation at site 2. It's very, very unlikely, unless the population is extremely large, that these two lucky strikes will hit the same individual. And so what happens is that now these two individuals who have beneficial mutations at two different sites, they cannot have sex and put their contributions together. And so therefore they are in competition with each other. And eventually it must be the case that everyone in the population is descended from either one or the other, which is a waste, because it's like that other mutation was good too, and it would have been good to have, for both of them to have reached 100% frequency. But that can't happen in an asexual population. One or the other must prevail. And then you have to wait and wait and wait for the good mutation at the other site to reappear again by mutation. And then you can see that the adaptation time in this asexual population is much longer than in a sexual population, where essentially both positive mutations can simultaneously increase to 100% frequency. So basically, that was Fisher's idea. And then another geneticist named Hermann Muller, who was a pretty insightful guy as well, basically came up with the same idea. And then later, Hill-Robertson interference came along. And then what happened was that in 1974, a big paper in genetics by Joe Felsenstein showed that all these different formulations are actually mathematically equivalent. And since then, they've all sort of been treated together. Yeah, Dalton.
Two questions. One is, you didn't talk this year about parasites.
Yeah.
The Red Queen hypothesis, or anything. Is that dead now in the literature? Or
no, it's not dead. It tends to be more favored by like non-mathematical, like ecologically minded people who study this question. I would say that the more mathematically minded people, the problem they have with it is that if you try to formalize, okay, so the Red Queen hypothesis basically says that, and this was actually an idea that William Hamilton liked a lot. I guess you could call him the originator of it. Basically, the idea is that the problem that large-bodied, long-lived organisms like us have is that we're constantly being parasitized by smaller, shorter-lived organisms. And these shorter-lived organisms, because of their shorter generation times, can adapt extremely effectively to take advantage of our cells, to reproduce for themselves, and to hurt our own reproduction. And so basically, the Red Queen hypothesis is that by essentially creating a unique barcode for each individual, each generation by sexual mixing. That then this encodes like a different combination of like receptors on the surfaces of cells and so on, or different idiosyncratic patterns of behavior, say, that like cause you to avoid parasites. And the idea is that that will keep you from just being totally taken over by the parasites. And the reason it's called the Red Queen is because in Lewis Carroll's book, Through the Looking Glass, this is a different character from the Queen of Hearts in Alice in Wonderland. The Red Queen basically tells Alice at one point, well, here you have to run as hard as you can just to stay in the same place. And so the idea is that, well, the mixing up of your genomes in one generation is not enough to get you to be able to escape the parasites, because then the parasites evolve, and then ensues this like constant pursuit in high-dimensional space. And so therefore, that's the Red Queen hypothesis. I would say that the problem that people have with that is that if you try to formalize this idea to a greater extent than Hamilton did, it turns out that this will sustain sexual reproduction only in special regions of the parameter space. And so therefore, people tend not to like it so much. But it's certainly not dead. There are pluralists who like to say, well, everything is working together. And there was actually a paper in Science a few years ago where it was another experimental evolution study where they have these different lineages of C. elegans, and they infect some with parasites, and others they leave them alone, and they see if some switch to sexuality and so on. And in this experiment, actually, it turned out that the lineages that are exposed to parasites switched to sexual reproduction and maintained it at higher levels than other lineages.
Can't you view the parasite story as a special case of the linkage disequilibrium story? And it seems like a form where you have the epistasis of the right functional form. So it may be only one of the many kinds of linkage disequilibrium, negative linkage disequilibrium stories with the right kind of functional form, but isn't the parasite story an example of that?
Yeah, I believe I've read about attempts to sort of, frame it in a way that it's kind of becomes equivalent to all the other things. But I mean, the paper that I assigned by Sharp and Otto, I think the main distinction they made was that I guess the one fundamental qualitative difference you can point to is that, like, the Red Queen hypothesis says that diversity is good inherently for its own sake. That is, if the population becomes genetically uniform, then it's doomed, and then rare types are favored, and that's why sex is good, because it essentially makes everybody unique. Whereas this emphasis on diversity being good for its own sake is not, I feel like it's not so present in the family of hypotheses that I stepped through here. Here it's more that like, there's a state of the world in those family of hypotheses where everybody is ++, for example, and we could somehow magically prevent deleterious mutations from happening, where it would be like, yay, we're great. But in the Red Queen hypotheses, it's like genetic uniformity is never good, and this constant high-dimensional pursuit has to just always happen.
But you need the environment to change often enough, or else the finite population story doesn't work. So you have to have, the thing about the parasites is they change often enough that the finite population thing has some bite to it. But if you didn't have, you have to have a changing environment for finite population doesn't have bite.
An infinite population would not need to worry about parasites.
I'm also saying a finite population with a totally stable environment can be asexual.
Well, the thing is that in the negative LD explanations, as opposed to the Red Queen explanations, usually it's just kind of assumed that bad mutations always happen and that they're unavoidable. And therefore, because we all carry varying numbers of these bad mutations, and some of them can even drift to pretty high frequency, becoming common variants, And then we have all this heritability of fitness, which is a problem that needs to be resolved. I guess the qualitative difference between the two families is that in the Red Queen world, there's no such, I mean, there's nothing in that theoretical world that says there are these unconditionally deleterious mutations. It's just the effect of the mutation is always dependent on, like, you know, where the parasites are at the current moment. Yeah, there's a question.
It's linked to what you were discussing, but I mean, I'm guessing you're not talking about, you know, because you're talking about LD, you're not talking about these extremely sort of private deleterious mutations, which perhaps won't survive across generations for it to accumulate in a healthy sort of manner. But I guess when I sort of think about it, you can think of loci which influence sort of reproduction as some loci which, regardless of the environment, would contribute things like, you know, loci that influence intrauterine environment and things like that. But there would also be loci which are very much environment dependent. How would that sort of influence the negative LD situation? Because, you know, it can sort of negatively affect reproduction or positively affect reproduction, and then environment can change, and it can be a neutral.
Okay. So the fact that basically what is fit now was not what was fit 100 years ago or 200 years ago. I mean, that's a tough difficulty in testing the negative LD hypothesis in humans, because I suppose if you didn't get the unequivocal result that, you know, distant sites on average are in very low levels of negative LD, you could just say, oh, well, you know, the environment changed and we're still on our way to the new equilibrium, and that's why we don't get the result. But I would say, again, there's an asymmetry there. It's like, if you don't get it, these ideas might still be right. However, if you do get it, there is negative LD, which there might be, even though there's been dramatic changes in what constitutes fitness in many human environments. But the thing is, things are always changing. New parasites appear, old predators die out, asteroids crash into the Earth, the dinosaurs get out of the way. I mean, things are always changing, and yet it seems that sex is always beneficial. So it's, so I think it's reasonable to hope that despite the undeniable changes in what makes you a fit human being, so to speak, that we might still be able to get it.